Hi. Have you thought about how we are able to survive despite the dangerous bacteria and viruses lurking everywhere? Today, we are going to take a look at our immune system, the guardian of our health. In summary, there are two types of immunity, innate and adaptive. Innate immunity consists of three things: physical barrier, immune cells, and chemicals. Adaptive immunity consists of humoral immunity and cell-mediated immunity. Let's take a journey with Mr. Bacteria together and learn about these different components of our immune system. One day, Mr. Bacteria decides to take a trip in the human body. First, he chooses the route to enter. He thinks, the stomach acid will kill me, so I won't go with food. Let's wait for a clumsy boy to cut his thumb and we will go from there. After entering the body, Mr. Bacteria encounters the second line of defense, immune cells. For instance, macrophages, dendritic cells, and neutrophils, which eat the bacteria, mast cells, which release cytokines to induce immune response. If Mr. Bacteria traveled with a lot of other bacteria and viruses, adaptive immunity will come into play. B cells travel in the circulatory system and identify the bacteria by its antigen. Antigen is a toxin which induces an immune response. Once the bacteria is identified, B cells will start secreting antibodies, which are blood proteins counteracting a specific antigen. Once encoded by antibodies, the bacteria is marked for destruction. It is also slowed down, making it easier for phagocytic cells to hunt it down. The antigen-presenting cells, such as macrophages and dendritic cells, eat the bacteria and present the antigen of bacteria on the surface molecule called MHC2 molecule, forming a complex. The antigen will be presented to a T helper cell. The T helper cell then travels around. When a T helper cell encounters a B cell presenting the same antigen, a large immune response will be activated. T cell and B cell will clone themselves and release a lot of antibodies and cytokines. If a virus successfully attacks a cell, that infected cell can ask for a mercy killing by presenting fragments of antigen on its surface. A cytotoxic T cell will then recognize the signal and induce apoptosis.
Now let's look at the functions and properties of all the cells involved in the innate immune system.
So, let's talk about dendritic cells. Many of you may have never heard of dendritic cells, but they are just inside your bodies, actually just underneath your skin. They are guarding your health all the time by keeping an eye for any potential intruders. They are the key antigen-presenting cells, which means they spot and catch antigens that enter your bodies and present them to T cells. So what are antigens? Basically, any foreign intruders like bacteria and viruses. Toxic chemicals can be antigens. Pollens can be antigens. We humans encounter those antigens all the time, and dendritic cells protect us. They lie and wait in our skin, underneath our skin, and one day you cut yourself in the arm and very unfortunately, the site of injury got infected, and now your body has to fight the invading pathogens. But lucky for you, dendritic cells bind with antigens and become activated. The activated dendritic cells start to migrate to lymph nodes through the lymphatic vessels. In the lymph nodes, the dendritic cells will meet another important player in our immune system, the T cells. When T cells recognize antigens that was bind to the surface of the dendritic cells, they become activated and start proliferation. Then they will continue our body's fight against the antigens that dendritic cells started and continue their role.
Next up, we're going to be talking about natural killer cells. Natural killer, or NK cells, are a type of lymphocyte that circulates throughout the body. These cells originate in various locations, most namely within bone marrow, lymph nodes, the spleen, tonsils, and the thymus. Once an NK cell matures, it gets released into the circulatory system to become part of the immune system. NK cells have two primary roles in the body. First, they are an important player in the innate immune system. This means NK cells are one of the first lines of defense against diseases, so bacteria, viruses, and even tumor cells. Additionally, their activity regulates the body's adaptive immune system, which means that they help teach the rest of the immune system how to react to any future invasions of the same type of disease. One method by which natural killer cells protect our body is through the direct killing of cells. So NK cells have a whole lot of receptors on the outside of their plasma membrane, some of which are inhibitory receptors and some of which are activating receptors. If the NK cell gets stronger activating signals, then it will trigger the direct killing response. This response results in a release of chemicals called perforins and granzymes. The perforins basically make pores in the targeted cell's membrane so that the granzymes can enter the target cell and trigger apoptosis, or programmed cell death.
Our immune system has a key component called the white blood cells, of which there are several different kinds of, and I will be discussing monocytes. Monocytes are a type of blood cell that fights off bacteria, viruses, and fungi. Monocytes are the biggest type of white blood cell in the immune system. Originally formed in the bone marrow, they are released into our blood and tissues. When certain germs enter the body, they quickly rush to the site for attack. Monocytes have several functions to help us ward off diseases and infections. To help us remember what monocytes do, note that the first letter of monocyte begins with M, for munch. Monocytes munch. Monocytes have the ability to change into another cell form called the macrophages before facing the germs. They can actually consume or munch on harmful bacteria, fungi, or viruses. The enzyme in the monocyte's body kill and break off the germs into pieces.
Up next, we're going to talk about macrophages, which, if you ask me, look a little bit like anteaters. But anyways, macrophages are a type of white blood cell produced by the differentiation of monocytes. These cells engulf and digest cellular debris and pathogens in a process called phagocytosis. They can also stimulate lymphocytes and other immune cells to respond to a specific pathogen. Because of this, they play roles in both the innate and adaptive immune systems. But how exactly do they work? When tissue is damaged or an infection is underway, monocytes are recruited to the area and differentiate into macrophages. Macrophages have pattern recognition receptors on their cell membrane, which bind to pathogen-associated molecular patterns, or PAMPs, as you can see here, and these alert the macrophage that a pathogen is nearby. The macrophage seeks out the pathogens and ingests them, as shown. These ingested phagosomes then fuse with the lysosome to form a phagolysosome. The enzymes within this organelle break down the pathogen, and the waste material is either expelled or assimilated into the macrophage. Because macrophages are so central to the immune system, they are found in nearly every tissue of the body.
White blood cells are commonly classified into two categories, granulocytes and agranulocytes. Granulocytes are special because when viewed under the microscope, you can see large, visible secretory vesicles. This portion will cover granulocytes. Granulocytes differentiate along with monocytes and mast cells from a granulocyte macrophage progenitor. Like most white blood cells, they develop in the bone marrow. There are three main granulocytes: neutrophils, eosinophils, and basophils. The three granulocytes are distinguished based on differences in the staining of the cells, which indicates that they have different chemistry and functionalities. Location-wise, granulocytes circulate in the blood for only a few hours prior to leaving the system and entering our body's connective tissue, where they'll live for a few days before being recycled. In comparison to the macrophage, who can live for a year in the connective tissue, the granulocyte's life is rather short. However, their impact is very large, as we will see. Neutrophils are the most common type of granulocyte, distinguished by their multi-lobed nucleus. Neutrophils can kill foreign invaders either by phagocytosis, which involves in taking and digesting them, or by releasing the granules, which contain cytotoxic chemicals. Neutrophils make up to 70% of white blood cells and are the most common type of white blood cell. The sign that your neutrophils are at work is the buildup of their debris, which we see as pus. Eosinophils are noted for destroying parasites via phagocytosis, as well as modulating allergic inflammation by secreting their granules, which contain antihistamines, which counter the inflammatory effect of histamines. Eosinophils are far fewer than neutrophils, only making up 4% of the white blood cell population. Basophils, in contrast to eosinophils, help increase inflammation by secreting histamine granules. Basophils are the least common of the granulocytes, making up less than 1% of the total white blood cell population in our body. Together, the three granulocytes play important roles in our body's immediate response to external threats via the innate immune system.
Now let's look at the functions and properties of all the cells in the adaptive immune system.
T cells are white blood cells, and therefore are derived from stem cells in the bone marrow. T cells are located in the blood and lymphatic system, including the spleen, lymph nodes, and tonsils. For this reason, T cells are also known as lymphocytes. The thymus plays a crucial role in the development and maturation of T cells. In the thymus, it is also where the cells become mainly differentiated before entering the circulatory and lymphatic system. T cells are mainly differentiated into two different types of cells: CD4 positive or T helper cells that coordinate and activate other immune cells, and CD8 positive or cytotoxic T cells that find and destroy infected cells. T cells are also critical to the adaptive immune system and become activated when the innate immune system fails to eliminate our enemies, the germs. But how do they undergo the attack? First, the pathogen is presented to the CD4 T cells by an antigen-presenting cell, usually a dendritic cell. These CD4 T cells then become activated and release chemical messengers in the form of cytokines to activate all the immune cells, including CD8 T cells. When the CD8 T cells recognize the pathogen, they release cytotoxins, which are toxic substances including perforins and granzymes that eventually kill the infected cell and stop the enemy from spreading.
Now let's talk about B cells. B cells are different from some of the other immune cells like T cells because B cells don't directly kill the pathogen. Instead, B cells are going to act as factories that produce antibodies which help the rest of the response. Let's dive really quick into how that works. Here you see a B cell with some spiky receptors sticking off the end. These receptors are specific to different pathogens, which have their own matching receptors sticking on the edge of their cell as well. Let's now imagine that the B cell encounters one of these antigens. Oh no, your body is under attack. This signals the B cell to duplicate itself a bunch of times and to mature into a plasma B cell, each of which can produce thousands of antibodies. All of a sudden, you have an army of antibodies to protect yourself. There's a catch to this though. I mentioned that the B cell needs a specific receptor to recognize the threat. Unfortunately, it can take days to train a B cell to produce the right kind of antibody. Thankfully, we only have to teach B cells once, because after you get sick, the B cells stick around and become memory B cells. The next time you get sick, these memory B cells are right there, ready to go, preventing the infection from getting out of control and making you sick. You can remember this because little kids are sick all the time. Why? Because they haven't built up this memory bank of B cells that adults have that prevent the adults from getting sick. Quick fun fact, you might think that B cells are named for the place they're made, the bone marrow, but actually they're named B because scientists first found them in the bursa of Fabricius, an organ only found in birds.